Hi, everybody. We're going to start the next session. If we can, I want to encourage the discussion after such a good last session, but if we can give the space to the speakers because they deserve the time. My name's Dane Bacano-Kelly. I'm a co-chair for the organizing committee today, and I will be chairing this particular session for the Flash Talks, the second of 3 Flash Talk sessions, at which it gives the speakers the chance to showcase their work and entice you to come and talk to them more at their posters, or just in general across the entirety of the particular event. Sorry, once again, can we have some space at the side, please? There we go. So we have 5 speakers for you today. They will be limited in their time as per a normal Flash Talk, so they'll have 3 minutes. As a result, we're only going to have 1 question per person at the end. Try to keep the question concise so that they can deliver it, and then accost them afterwards to tell you more detail about it. That being said, please do ask questions, because these guys have worked so very hard at giving their talks and putting it together. I'm sure they would welcome the opportunity to be spoken to and asked about their work. I know I would. And also, there will be a roving mic, so just pop your hand up and the person will come through to you. So, I think, seeing as we're running late, without much further ado, I will invite the first speaker, Jacek Szymański, who is going to be giving us a talk on unraveling the role of BDNF TREX signaling in hippocampal circuit function by multi-omic holistic approaches.
Hello. Thank you. Thank you for the invitation. My name is Jacek Szymański, and today I will tell you about how multi-omic technologies are helping us uncover the critical role that BDNF TREX signaling plays in shaping the hippocampus. So, first of all, the dentate gyrus is the gateway into the hippocampus. It receives input from the entorhinal cortex, passes it along the mossy fibers to the CA3 region, and further along the Schaffer collaterals to the CA1 region. And if we look into the dentate gyrus, the dentate granule cells start off as really glia-like cells. And as they mature, they progress through stages of like neuroblast and immature stages to become fully integrated neurons. And as they do that, they migrate from the subgranular zone into the granule cell layer. And in our case, we have selectively deleted TRAC-B from the immature dentate granule cells. And this led to delayed migration, maturation, and integration of these cells into the granule cell layer. And not only that, it resulted in a disrupted communication within the trisynaptic circuit in general, leading to altered long-term potentiation, premature GABA shift, and eventually impaired cognitive processes. So we wanted to have a look at what happens at the molecular level. For that, we used paired single nucleus RNA and ATAC-seq to have a look both at the transcriptomic and the epigenetic data simultaneously. And what we found was over 1,000 differentially expressed genes and more than 300 differentially accessible transcription factor motifs. And interestingly, some of those were actually in stages before TRAC-B was deleted, which hints at a more broader aberrant bidirectional signaling. And so, in general, our study not only helps us better understand how early signaling imbalances may cause neurodevelopmental disorders, but also points us towards very specific therapeutic targets that we can use to alleviate them. And with that, I would like to thank you very much for your attention. Thank you very much everybody who was involved in the study. And please visit my poster PT 175 for more details. Thank you very much.
Thank you, Jacek, for keeping so well to time. Questions? Question? Give you a second to formulate it if you'd like.
Thank you. That was really interesting. I was wondering if you could just comment maybe on some of the.
Sorry, can you a bit closer because I
Oh
can.
God, it feels very loud to me.
Oh, sorry. Oh yeah, it's
behind.
pointed that way.
I was wondering if you could just comment on some of the dysregulated pathways specifically, or any bit more on the mechanism of all the differentially expressed genes and transcription factors.
As in, certainly. Of course, most of the pathways that are dysregulated are dealing with development of the cells. So that's the main point. There is, for example, CREB is dysregulated, and for the rest of the more specific details, we will soon be possibly printing a preprint, and then as in I'm not fully at liberty to disclose all the as when it comes to the pathways. Let's just keep it at developmental.
Thank you very much. Another round of applause, please, I think. So our second speaker today will be Yassine Malik, who will be presenting another TrkB orientated talk, conditional TrkB deletion from striatopallidyl neurons in mice, and leading to early age hyperdopaminergia preceding hyperkinetic dysfunction. Take it away.
Hello everyone, I'm Yassine. So I will be giving you a quick snapshot of my poster while I discuss the role of BDNF TrkB signaling in the development of hyperkinetic disorders like Huntington disease. So Huntington disease is a very fatal neurodegenerative disorder with no cure in sight. The reason being the complex pathogenesis of the disease. This particularly disorder starts with the degeneration of indirect pathway neurons, which we will call ISPNs. Also, the role of dopamine is not clear in this disease. So one of the pathways that's implicated in the early Huntington disease is the impairment of BDNF TrkB signaling. So what we did in this study was to study the role of BDNF TrkB signaling in these neurons, which we call ISPNs. For some reason, the animation isn't working properly. So the deletion of the TrkB in ISPNs, it leads to development of an age-dependent hyperlocomotor disorder. So this hyperlocomotor phenotype was triggered by an early age hyperdopaminergia. So what leads to the hyperdopaminergia is we found that there's a neurotransmitter switch in the dopaminergic cells in the midbrain, which leads to switching of low dopamine producing cells into high dopamine producing cells. Further, we did transcriptomic analysis and functional validation. So what we see is that there's a gene called GST02 gene that precedes and triggers the hypodopaminergia. So the GST02 has a function of dehydroascorbic reductase activity, which recycles ascorbic acid and is responsible for the overexpression of tyrosine hydroxylase and production of dopamine in the substantia nigra. So what does GST02 upregulation and dopamine upregulation together do is this leads to glutathione redox system imbalances as well as a deficit of energy metabolism systems like OXPHOS and electron transport system. So the question arises is if we reduce the GST02, does this rescue all these phenotypes that we see? The answer is yes. So what we do is we selectively reduce the GST02 in these ISPNs. So we rescue the hyperdopaminergic phenotype. We rescue the energy metabolism deficits. We also rescue the development of the early age locomotor deficits as well as the later age hyperlocomotor phenotype. So this was the first time that a relationship was established between glutathione ascorbic metabolism, BDNF TrkB signaling, and the development of hyperdopaminergia in a neurodegenerative disorder. So feel free to read my paper as well as to see my poster at 003.
Perfectly timed, right on time. Questions? Oh, sorry. Fair enough.
I did my PhD on BDNF and Huntington's disease, so thank you. Did you have a look at what was going on with the dendritic spines in the striatum in your models? Because one of the things we'd seen was that BDNF released from the cortex was influencing the spines in the striatum in Huntington's models. Did you look in your model?
In this study, we didn't look at anything about spines, but previously we have looked at the spine density. So the loss of TrkB leads to reduced density of the spine. So that has been shown in like a previous study. Yes.
Thank you very much.
Thank you.
Our next speaker is Konstantin Yulian Chita. Oh, perfect. I apologize, didn't vote advance. He's going to be presenting on short-term face familiarity displays greater modulatory effects for neutral expression than for emotional ones. I'm going to leave it there. Have we? Yeah, you've got it on the screen now, I think. Or it's cycling through.
Thank you. Hi everyone, my name is Konstantin, and I'm a face recognition and emotion processing researcher. And today I will tell you a bit about one of the studies that we have conducted as part of my PhD. So face recognition is an important societal and overall evolutionary process because we can extract information related to someone's age, gender, or ethnicity, or even we can infer personality traits such as aggressiveness or trustworthiness simply by looking at someone's face. Now, it is because of this high level of informational content that we can extract that face recognition became such an important and widely researched topic in the literature. And it became such a popular topic, actually, that we have too many papers at the minute that tend to cause some controversies. And one of these... Thank you. And one of these controversies actually is presented in the first image. This is the model of face recognition described by Duchesne-Novel in 2015. And if you can see over here, we have a lot of brain regions that seem to be interconnected in the early processing stages, and then they tend to become more and more independent the further the information travels along the relevant neural pathways. And if we were to simplify all of these brain regions, we would have at the very top one neural pathway that is involved in facial expression processing, and at the bottom, a pathway that is involved in feature processing. Now, in our experiment, we wanted to test this level of interconnectivity, and to do so, we needed stimuli that were able to activate both pathways simultaneously. And this is why we have used 36 triplets of identities depicting happy, neutral, and angry expressions, and we have morphed their facial features in 3 different intervals, 25, 50, and 75% similarity, with other faces that were matched in terms of age, gender, and ethnicity with the initial identities. And then we have asked our participants to provide similarity ratings for the unmorphed emotional faces and the morphed faces. And we have collected EEG data and reaction times, and we have found out that people did respond faster to neutral expressions compared to happy and angry expressions. And they also tended to provide higher similarity ratings for the neutral than happy and emotional expressions as well. So we had a very nice matching set of results over there. In terms of the EEG data, however, the picture is not very clear. We did find that all of the emotions were processed kind of around the same at the same time and with the same intensity. So these results do indicate that even though we do process everything at the same time, there is a discrepancy somewhere that is modulated by familiarity that we are trying to currently explore in further studies. Thank you.
Thank you very much. Yeah, we've got a lot to compete with noise-wise, so apologies for having to switch over halfway through. Questions? No? Yeah. Okay.
Oh my God, thank you. Very interesting work. Do you not get an age positivity bias with your data, which means that as people get older, they rate emotions more positively?
Try to account for this as much as possible. We have used stimuli that were as homogeneous as possible. We have run a pilot beforehand to ensure that all of the emotional expressions had the same valence, the same intensity, and people were able to recognize everything as equal as possible in terms of emotion. So I would not necessarily expect there to be any sort of positivity bias, but obviously further research that is more focused on this could indicate whether there is something there or not.
Thank you very much.
Thank you.
before you come up, if there are any further issues, just signal, gesture to me. I'll hand you this one, unless you would prefer to work with this one in the first place. We're competing with a lot of noise, so it's pretty hard. So the next speaker will be Rebecca Woods, who will be talking to us about linking early life environment methylation of immune and stress response genes and child mental health outcomes in a longitudinal human birth cohort. Do you want to try that, Mike? And we can.
Can everyone hear me? I get this close? Brilliant. So yeah, my work all focuses on trying to link that early life environment, epigenetics, and mental health outcomes in children. So I'm sure most people are quite familiar that early life stress is a risk factor for increased poor mental health outcomes in exposed children. But one of the things I'm trying to understand is the mechanisms that link these two events, with a key hypothesis being that the early life stress leads to altered epigenetic regulation of immune and stress response pathways, and it's this that predisposes individuals to neuropathology. One of the ways in which I'm investigating this is through the work in a longitudinal human birth cohort known as the World Child Health and Development Study, or WCHADS, which is a partnership between the University of Liverpool and MMU. So the way this study was set up is we recruited women during their 20-week scan, and we subsequently followed these women throughout their pregnancy and have continued to follow the children since, with this study still very much ongoing. I would like to point out, for the purpose of this talk, I'm only focusing on the developmental window indicated there. But some of the things we've done is to perform early life stress measures, including maternal measures, home life measures, and then we've looked at the mental health outcomes in these children through various behavioural tests done at clinic visits, either yearly or bi-yearly. And during these clinic visits, we've also been able to take saliva samples to extract DNA and look at epigenetic changes. So for the purpose of the results here, the early life stress measures I've looked at initially were prenatal and postnatal maternal stress, maternal depression measured by the Edinburgh Depression Scale, and the child mental health outcome here was 3.5-year presentation of externalizing behaviors. My epigenetic measure I'm focusing on is methylation of the IL-6 gene, which is known to link with early life stress. So kind of exactly as we expected initially, both prenatal and postnatal maternal depression significantly associated with increased risk of child externalizing behaviors, showing that link between early life stress and poor mental health outcomes. So for the epigenetics, quite interestingly, postnatal but not prenatal maternal depression significantly reduced methylation of the IL-6 gene in the children, and this was significant from the 2.5-year time point and continued into 3.5 years. And then continuing on from that, what we were able to show is that that reduced IL-6 methylation in the children was also a significant predictor of increased child externalizing behaviors in these children. So together then, these results begin to build this picture where what we have is this early life stress exposure, altered epigenetic regulation of inflammatory responses, here the IL-6 gene, and then this increased risk of poor mental health outcomes in the externalizing behaviors. To hear more about the data that we're generating as part of this birth cohort, feel free to come and see me at my poster after, or ask any questions now.
Wonderful. Thank you very much for keeping to time. Questions? So you're using the IL-6 as a marker. It's obviously only one marker that you've mentioned today. Do you have others to make sure that it's validity, and how are those sort of worked out?
Yeah, at the moment we've profiled 12 genes, and we have done that across the entire timeline. So as I say, it's still ongoing, so we have that data up to 9 years. The biggest challenge we have now is doing all the analysis on the data that we're continually generating. But all the genes were selected as when we wrote the MRC grant with some validity to already being linked between early life stress and those outcomes. So in some way, be it through expression or through measures in autism, for example, and that's how we selected them.
So I primed all of these. That's why they're keeping to time so well. I primed them saying that if they were not on time, I'd start taking off items of clothing to just start to make them very worried. But our next speaker is Kavishini Aspamsi. Basami, sorry, apology. Examining hippocampal cerebral functional. We don't have anything on the screen. I'm going to speak very slowly while you sort that out. Examining hippocampal cerebellar function, connectivity across lifespan. Perfect timing. There you go.
Okay, well, never mind. So,
Stop the timer again. It keeps going off the screen.
okay, thanks.
1, 2, 3, go.
Okay, thank you. So I want to thank you for the introduction, and I wanted to speak about the first project of my PhD, which looks at hippocampus-cerebellar functional connectivity across the lifespan in humans. But I wanted to give you an introduction. So the hippocampus and cerebellum are thought to be distinct brain areas doing distinct processes in the brain. The hippocampus involved in memory and spatial navigation, and cerebellum involved in the automation of motor and cognitive operations. There is also evidence from rodents showing that these two structures interact with each other. So disrupting cerebellar neurons in transgenic mice affects hippocampal place cells firing and negatively affects navigation behavior. Evidence from lesion and tracing study, again in rodents, shows that they might have a direct or indirect connection, so suggesting that they have a structural connection. And both structures are thought to undergo similar age-related changes, where changes in cerebellar volume is linked to poor performance in hippocampal-dependent tasks. We wanted to find out whether the hippocampus and cerebellum, how is it topographically organized in the human brain, and how does it change during aging. So to do this, we applied seed-based connectivity analysis to the CAMCAN dataset, which had a large age range. And for ease, what we did, we took 4 hippocampal seeds, left, right, anterior and posterior, and connected to the whole brain in the contour box, and then connected for family-wise error, and then laid the results on cerebellar flat maps. So what we found was a distinct pattern of connectivity. So the left hippocampal was preferentially connected to cross-2, the right hippocampal preferentially connected to the left cross-2. And as you may be aware, there is a long-axis functional gradient along the hippocampus, and this reflects this connectivity pattern that we found. So the anterior hippocampus is preferentially connected to cross-2, both regions thought to process higher order information. Posterior hippocampus was preferentially connected to lobule 5. Again, both regions are thought to be involved in kind of sensory motor processing. For aging, we saw that there was a reduction in functional connectivity during aging between the left, right, anterior hippocampus and the border of lobule 5 and 6. And we saw minimal results for the posterior hippocampus. Future studies will investigate the importance of this connection during spatial navigation, and this is presented at poster PT091. I would like to thank my supervisory team for the support on this project. There is a preprint available if you want to find out more, or I'll be at this poster presenting this later on.
Thank you very much. Wonderful. Questions? It's done in silence again. What were the ages that you actually selected, and why did you specifically select those ages for mapping those? Why were they key and critical?
So the ages that I used was 18 to 87, and this was because that what was available in CAMCAN. So it was the easiest way to kind of select those ages. Yeah. No worries.
So that actually brings us to a close for our flash talks. Please go and speak to the speakers. They will be ready and waiting in this corner and then running towards their posters, I think, to go and present. But they've given you their poster talks titles, so please, they've worked so hard on this. And another round of applause for all of them. Thank you.